Thanks a lot for this very nice introduction, as usual. And I'm impressed by the level you already put in that. So you've been explaining quite a few things already. That's excellent. And so thanks also for everybody at Radcliffe, all the staff, everybody there. It's really a wonderful place to have ideas, to rest in, change our minds. It has been a great time for me so far, and I'm really looking forward to it the coming year. So I'm here in Radcliffe to work on the topic of gene regulation and gene expression. It's a very broad topic, vast topic that touches on very many things in biology. I will just take one angle today just to give you one example of my research in this area today. So first, I'd like to start by acknowledgements. Some of the work I've been presenting will be have been done in collaboration with Frédéric Fion, who was a PhD in my lab. He's now finished his PhD. And much of what I will be talking about is taken from that PhD and some other work with some collaborator mentioned here. So today, my talk here to you is you've got to take it like a dessert, right? It's we are here to understand new things, perhaps. I know that most of you are not biologists, so I will have to provide a lot of background material for you to understand my point. So what I can promise is that it will be a certified organic evolution, that dessert, that it will be completely free, of course, of creation-modified ideas. And, but, you know, there will be real bits of theory into that talk. And so it might become at time a little more difficult, so I will warn you when it is the case. And it's a new flavor. You know, it's some new research, something that has not been presented so far, or in different parts. So, one of an important puzzle in modern biology is the question why eukaryotic gene regulation is so overwired. So I will explain many of those terms. Here on that picture, you see the gene network and the regulatory networks of the genes in yeast that have been depicted with the different cellular functions in different colors. And it's really a huge complexity, and it's very difficult to not only to understand what's going on, but also to understand how it came about in the first place. Why is it that you've got so complex regulatory networks in eukaryotes especially? So that's my question today. And the answer is, and all my talk is just in that slide. So I will just spend my talk to explain this slide to you. So it says that in prokaryotes, there are haploids. I will explain the terms. Just you got an overview now. And you got simple regulation. In eukaryotes, you've got meiotic sex and recombination, something I'm happy that Judy mentioned in the introduction. And you've got also diploidy, and therefore complex regulation. So the red arrows here are intended to mean a causal relationship. And so I won't explain all those arrows today. I will mainly explain this one and this one. And I will, of course, give you some background to understand what it's all about. So we got to start with that background part about what natural selection is all about. And you probably know, most of you, I'm sure, know about Darwinian evolution and about natural selection. The principle of natural selection is pretty simple. When you want to understand why a trait has evolved, then you got to understand three things: that you got trait variation, which is brought by mutation, as these traits variation is heritable and inherited through generations, and that you got differential reproduction or survival of individuals carrying those mutations. And that's it. That's it. That's pretty simple. That's just the core of Darwinian theory. And here, you've got the Gary-Larson explanation of it, that short-sightedness is probably not something you would expect to evolve. So that's the first principle, and you know, everybody, I'm sure, know about it. The second principle is that, you know, perhaps on longer time scales, like on what we call macroevolution, you got some other stuff going on, right? Perhaps rare events mess things up, like, you know, the extinction of dinosaurs and things like that. So here, Gary Larson is perhaps not as good in the explanation, but you get the point. So that's most people, non-biologists, not evolutionary biologists, that's what, you know, they would see what evolution theory is about, right? And you may wonder, well, is that all? You know, it looks pretty simple. You've got very, very little principle, and yet you've got to explain biodiversity, you know, you've got so much to explain. All these life forms, is it possible to predict them, and all this diversity and complexity just from this very simple principle? And in fact, here I quote a very famous philosopher who had this quote about this. So Wittgenstein had this quote in a correspondence to a friend: "I have always thought that Darwin was wrong. His theory doesn't account for all of this variety of species. It hasn't the necessary multiplicity." And in some sense, you may, you know, argue with Wittgenstein about this, and I will do that. But, you know, it looks difficult perhaps at first sight that a very simple process like natural selection would give rise to complexity or diversity. Then, of course, natural evolution is not just about those small processes of natural selection. It's also a big historical thing, right? So that's the history of Earth from here and life on Earth. It takes like billions of years. You've got many, many events. And so the question is it really possible to build an evolutionary theory that would account for that? And, you know, from such simple principle, you might think, well, perhaps it's a difficult task. And in fact, this problem probably expands beyond, you know, evolutionary biology. Is it possible to have to make a theory about history? I know you've got historians in the audience, and it's a big question, you know, whether you could have a theory about history or just to explain things, or perhaps just see history as Shakespeare did in Macbeth, if life is just a tale told by an idiot full of sound and fury signifying nothing, just trying to emphasize the idea that history is just a matter of chance of many events, but there's not much to explain beyond, you know, that. And so these are very important critiques of evolutionary theory. And but at the same time, so we will come to those points as you will see. So the question here are: is it possible to build an historical theory? Is it possible to understand events in the very deep past? Can evolutionary theory explain multiplicity or complexity? And in fact, many people have been working on that. And you know that when you talk about this in general, you talk about major transitions. Major transitions here are pictured again by Gary Larson. He's a very useful cartoonist, where you see, you know, when you got evolution out of water. And that's probably the kind of transition people think about when you think major transitions. But they are not the transitions that are really difficult to understand, or the transitions that people are working on in evolutionary biology today. You know, they are not very theoretically challenging. In fact, the major transition in evolution were put forward by this book, a very famous book in the field, about 20 years ago, trying to explain what are those big transitions that we need to explain in evolution. And you got different kinds. The first type of transitions are about the becoming of groups. So when you go from solitary individuals to new higher level individuals through group formation. For instance, or you go from a unicell to a multicellular organism. This type of change. That's not the type of change I will be talking about. The second type of big transition is about energy, how you switch to huge different kinds of energy through the history of Earth. And the last type of transition is about genetic systems. That's the type of thing I'm working on. And this type of transition involves things like sex, recombination, meiosis, diploidy. I will explain some of those terms. And the central dogma. The central dogma, you perhaps heard about it. It was put forward by Crick. So the discovery of DNA. And the logic is that information flow in biological system from DNA to RNA to proteins. That's the blue arrow here. And this central dogma is about, this flow of information. And you may ask, how did that structure, that genetic system came about in the first place? And that's where the topic of gene regulation comes here, okay? Because gene regulation is, so the genes are on the DNA, and regulation, because they produce, you got this protein production, and that's how you regulate those steps. That's what is gene regulation and gene expression. So that's my topic today. And as you will see, it's connected to the other problem for genetic systems. So briefly, so that we can see where we are in history. So we are like 2 billion years ago, like the origin of eukaryotes. Eukaryotes, that's cells with nuclei, is about here. Eukaryotes are pretty much everything you know, every big organisms you know, plants, fungi, animals, everything but microbes or eukaryotes. And so we are here interested in understanding the evolution of gene regulation in eukaryotes in comparison to in prokaryotes. Of course, the origin of eukaryote itself is a very complex subject. I will not be going into that. So that's a prokaryotic cell and a eukaryotic cell. They differ in many, many ways. So you've got kind of a basic type of list of differences between prokaryotes and eukaryotes about their DNA, naked or not, circular or linear, but the presence of organelles, mitochondria in particular. The reproduction is very different. And that's something I would like you to remember. So in eukaryotes, you've got meiosis. I will explain in a minute. And eukaryotes also are diploids, which is not the case in prokaryotes. So that's the typical life cycle of a eukaryote. You got, that's how sex works in eukaryotes. You got two events, meiosis and fertilization. So that's the two key events that you find in all eukaryotic life cycles. And meiosis, so you got these two events, key events, separate the life cycle into two parts, the haploid phase and the diploid phase. So in the diploid, so we are all diploids in this room, we have two copies of each gene, right? That's one from your father, one from your mother. You are diploid. So that's the individual in this phase. Then you've got this phase here where the gametes are haploid, right? Little warning, here at meiosis, you've got this process that Judy mentioned, very important, that you've got recombination at that step. At that step, when gametes are produced, then the chromosomes cross over to produce a diversity of combination that is called recombination. So the next important difference between prokaryote and eukaryote is that their gene regulation is very different. Well, this one is pretty simple. This one is very complex. So how complex it is? So that's how it works. So you got here a eukaryotic cell. That's the nucleus. You got the DNA. The DNA is tightly packed by proteins, histones. Then it opens up. Then you got what is called transcription, where you got some RNA produced, which is not matured yet. Then you've got RNA processing, export into the cytoplasm. Then this RNA is translated into protein. And you've got a polypeptide here, which then becomes a protein after post-translational modifications. At each of those steps, you can have regulation of gene expression. Each of those steps, by various mechanisms. I won't have time to, to detail all of them. Here, I will mostly focus on the initiation of transcription, the major step here where you go from DNA to RNA. So it looks like this, in the cell. So imagine this is a chromosome, so that's DNA. You got a gene here. Next to that gene, you've got a regulatory sequence. It's called a promoter. At that site, you've got an enzyme which is called the RNA polymerase. It's just an enzyme that will produce the RNA from the DNA template. And the way this enzyme is attracted to that place really depends on a lot of other proteins and factors, that gather here and make a complex. And this complex, in fact, depends on what is called enhancers, other location a little further away on the chromosome that controls the attraction to those cofactors. And basically what differs for the gene regulation for gene A or gene B is that the identity, the sequence of those enhancers differ mostly. The next thing you've got to understand about gene regulation in eukaryotes is that you've got two kinds, what is called the cis-regulation and the trans-regulation. So that you are in a diploid, you've got this gene, which is in 2 copies. So in yellow here, you got the cis-acting sequence, like the promoter or the enhancer, that will control the expression level of that gene. And the control will be in part due to transacting factor, that is molecules that float around in the nucleus that will bind to those sequence. But the important point is that if you got a molecule floating around, it can bind here, it can also bind here. So transacting factors, they act on both homologs, on both chromosomes, whereas cis-acting factors, they just only act on the gene they are sitting next to. That's the only, but very important difference between the two things. So now you know more or less what is a prokaryote, what is an eukaryote, that the eukaryote have meiotic sex. So we can start, right? That was the introduction. So I will start by explaining this arrow. Why from when you got diploid, got meiotic sex, you have Diploidy evolving. So why are we diploids? Before I get to that point, you've got to understand a very important feature of diploid genetics and diploid. So in diploids, in haploids as well, but when you've got mutations, most mutations are deleterious, and we all have a lot of deleterious mutation. Every organism carries a load of deleterious mutation. And it's very important to understand that when a mutation occurs, then let's say you got an allele of the gene which is big A, then let's say you got a deleterious mutation little a arising in the population. So you've got 3 types of individuals, the little a, big A, and big A. That's called the heterozygotes and the homozygotes. And so that's the 3 types of genotypes. And here, if you represent fitness, so the fitness is a measure of evolutionary success, then you can say that as a reference, the big A has a fitness of 1, and that the presence of the deleterious allele at that gene decreases the fitness of that individual by a quantity S when it is homozygote. So when you are in a heterozygote individual, the effect of the mutation is then 1 minus HS, and H is called the dominance of that mutation. And in fact, dominance of deleterious mutation have been measured in a huge variety of organisms and cases and situations and genes. And deleterious mutation tend to be recessive, meaning that H is below 1/2. And in fact, it's about 0.25, 1/4, on average, across many studies. So the presence of those deleterious mutations in population cause what is called a genetic load. So in an haploid population, imagine you've got deleterious mutation occurring at a rate U. Then you've got these two alleles, big A and little a. This one has an effect on fitness of 1 minus S, as was just explained. In diploids, you've got twice as many mutations because you've got twice as much DNA. And you got the fitness I've just explained in the previous slide. So now you can compute what the frequency of the deleterious mutation should be in the population. And well, as an approximation, the frequency of the deleterious mutation is about U over S in an haploid population, and is about 2U over HS. H is the dominant in the diploid population. So it means that that's the frequency at which you find those mutations. Now you may want to ask, but what reduction of fitness overall, on average in the population, is caused by the presence of those deleterious mutations? So, well, that's just the frequency of those mutations times their effect. So if you multiply their effect, and here they are mostly found as heterozygote because they are rare, you find these very famous results. In fact, it goes back to almost a century now. That's the genetic load, meaning the reduction, the fitness reduction in a population due to deleterious mutation just depend on the mutation rate. And is twice as big in diploids because you got twice as many genes. So it is not dependent on the intensity of selection on those deleterious alleles. So because this load is twice as large in diploids, right? Meaning that the average fitness of diploids should be lower all the time than the average fitness of haploids. You may think, well, why did diploids evolve in the first place if their average fitness is lower than haploids? Well, that's a difficult problem. And in fact, to understand what's going on, we've got to turn to something you probably know, that story about the selfish gene. You probably heard about it. There's an idea popularized by Richard Dawkins, just meaning that to understand what's going on in evolution, you've got to think about not only in terms of individuals, like the usual Darwinian way is all about, but also in terms of genes. And so that's just a way to look at things. There is no difference. And it may offer clearer insights, but it's just a matter of perspective. And so we're going to practice that this afternoon a little bit. So to practice, we make a little model. So imagine you've got this same gene as before with the same fitness effect, the same story. And now you think that there is another gene. And this other gene changes in the life cycle how much time you spend in the diploid or in the haploid phase. That's all it does. And you wonder, will that gene increase in frequency? Or will it decrease in frequency in the population? Meaning, will it evolve, or diploidy will evolve, or haploidy will evolve? That's the way you solve it. You solve that problem. If you do that, so you got this two loci model, and they recombine. You got a recombination between the two genes at a rate R. So when you analyze this model, that's the result. That's the recombination rate between the two loci, and that's the dominance of the mutation. I told you that the dominance of the deleterious mutation is about 1/4, so about here. And so if recombination is quite high, so that is if you are in this red area, and that's where most eukaryotic organisms are, you'd expect diploidy to evolve, despite the fact that it reduces mean fitness in the population. That's perhaps there is a trick. So what's the trick? So I can put up an equation. Of course, it won't explain you the trick. So I will try to use metaphors, of course, to explain you the result. So the first metaphor is a metaphor, you got a double-edged sword. You got two problems here. The first problem, the first edge, is that it's good to mask deleterious mutation. You know, when you are in a diploid, you don't feel, the deleterious mutation as hard just because they are masked, because they are partially recessive. So perhaps it's a good thing. At the same time, if these mutations are masked and hidden to selection, they will accumulate in the population. So perhaps it's not such a good thing. But, you got to weigh those two ideas. So how do you do? Well, that's where the second metaphor comes in. And that's where we try to practice the selfish gene idea. So imagine for a minute, you are a selfish gene causing a little bit more of diploidy, and you arrive in a population where you got only haploids. What's going on? Well, first, you enter a population because you got haploids where deleterious mutations are very efficiently purged out of the population, so it's quite a nice place to be. There are not so many deleterious mutations around. And if you are unlucky enough to have to be associated with such a deleterious mutation, it's not a big deal either, because precisely you are a diploid. You are in a diploid individual because you are a gene causing diploidy. As a consequence, you don't feel that the effect of that mutation too hard. So it's not such a bad thing. But this mutation will not be weeded out of the population because you are a diploid, right? The trick is that if recombination is large enough, this deleterious mutation, which is still around, it was not eliminated in the population, will be a burden to everyone in the population, not only you as a gene causing diploidy. So what's going on in fact? Because recombination, reassort genes. So what's going on is that you enjoy the benefit of masking that deleterious mutation, but you share the cost with everybody else in the population. So it's no surprise that such a strategy evolves. Well, that was the hard part of the talk. There is another part like that. So that's where you perhaps get the idea what this selfish gene idea is all about. You start thinking a little bit more clearly about what's going on when you take the perspective of a gene doing this or that, even if it's just a device that you use to make things clear. Of course, the third metaphor here is that natural selection here is not optimizing average fitness in the population. So on average, the fitness in the population across all individuals is reduced by natural selection. So if you like the invisible end of natural selection, to take an economical metaphor, is not maximizing average fitness. And that's something that you got to remember about most situations about evolution. It's not so simple. Summary of this first part. If there is recombination in the population, if deleterious mutations are at least partially recessive, then it follows that diploidy is favored, meaning that if you consider a gene that extends diploidy, it will prevail. And this evolution leads to a lower average fitness of the population, but that's a side effect. So, when you got recombination, you evolve diploidy. So now we'll spend the last of the part of the talk to explain you the relationship between diploidy and complex regulation. So the evolution of gene regulation in diploids. So we go back to diploid expression. So here again, we've got a gene with two alleles with deleterious mutation. We've got a cis-acting regulator in yellow and a trans-acting factor, and they contribute to produce proteins, some of which are green because they are produced from that allele, some are blue because they are produced from that allele. And they are kind of roughly produced in the same amount because they've got the same regulatory sequence, okay, in cis. Now, what's going on if you got mutation in this cis regulatory sequence? Imagine you've got a cis acting factor which is a little bit more efficient at attracting the transcription factors, like this, or here. If this stronger, let's call it stronger, cis-regulator is close to the allele little a, then you will produce more of little a protein, more of the green protein. If this cis-regulator is stronger, the stronger cis-regulator is close to the big A, then you will produce more of the blue protein, and not 50-50. And in fact, when people go out and measure in using transcriptomics, for instance, how much you've got imbalance between expression in allelic expression, you find a lot. So that's an example where quite old studies, early days of transcriptomics, where you got nearly half of the genes, where you got more than a twofold difference in expression level between the two homologous alleles. So now you've got to think about the same thing, but in terms of what are the consequences on fitness for the individual. So imagine, so as before, that the little a is a deleterious mutation. Then if you produce more of that deleterious mutation, of course, the fitness will be reduced. And if you produce less of it, then it will be a little higher. So in effect, variation in the cis-regulator will cause a variation in the dominance of that mutation. The dominance here will be a little bit higher, and here it will be a little bit lower. So you want to ask, well, if you've got mutation on those sequences, then you will have allele-specific expression, then you will have changes in the dominance. So what are the evolutionary consequences of that? So we go back to this expression to fitness graph, where here imagine you got the fraction of expression of this little a. So where you were in standard heterozygote, you produce 50-50 of each protein. Then you've got the same as before, dominance of H. If the stronger cis-regulator is close to the deleterious mutation, then you will produce a little bit more of that protein, and therefore the fitness will be a little bit more reduced because you got more of that deleterious mutation, that defective protein, sorry. So that's the two value. Because this function is concave, you got this property, which is inevitable, that you got the average of those two dominants here will be larger than the H values you started with. That is a detail. You will see later, perhaps. And next, you do a model, as we did for the Diploidy case. You put exactly the same ingredient. You got deleterious mutation, then you got the fitness, and you introduce a new allele that causes a little bit of more of expression or less, and you see what's going on, what's the outcome. So if you look at the outcome for a weaker cis-regulator, that's the result. So that's the log recombination. That's the distance between this regulator and the gene in log, so meaning that it's very close to the gene here, and it's quite far here. And that's the neutral expectation. So that's a way to measure evolutionary success. You compare the fate of an allele that you introduce in a population in one copy to the probability of fixation of an allele which is neutral, it does nothing. And you compare the two. If the fixation is lower than neutral, it means it's selected against. When it's above neutrality, it means it's favored. So as you can see here, a cis-regulator is always weaker. A weaker cis-regulator is always disfavored. In contrast, a stronger cis-regulator is always favored, except when it's too far away from the gene in terms of recombination here. So stronger enhancers, stronger cis-regulator are always favored if they are close enough to their gene that they regulate. Of course, you've got a lot of models you can do about this. And again, the metaphorical explanation of what's going on is simply that you got, as in the Diploidy model, you've got two effects, one of masking and one of purging. The masking effect is always, so it's always a bad thing to have a new cis-regulator in the population because it increases average dominance. Deleterious mutations are more exposed, are more visible to selection. It reduces mean fitness of individual. But for stronger enhancers, stronger cis-regulator, the gene that sits next to them will be more visible to selection, more exposed to selection, and therefore better purged from deleterious mutation. And so on average, those stronger regulators will find themselves always on good genetic background, if you like. And that's the reason they spread. And in fact, they spread indefinitely in this model. There is what is called a runaway. There is no reason, except if you put it in the model, that this process will stop. It goes forever. This regulator go further, the increase in strength further and further and further. There is no limit, at least in the model. But it's like you run and you stay at the same place, because diploidy did not change, dominance did not change. After the spread of a stronger cis-regulator, you are back to the same situation where you got 50-50 expression. Deleterious mutation have the same effect. Everything's the same, except that you got constant evolution on this cis-regulatory region. And of course, the strength of cis-regulator, you can think in it can be different things. It can be that the sequence where you got the matching between the cis-regulatory region and the transcription factor coevolve. It can also be that you got a duplication of those sequences, or that these sequences start to attract different transcription factors just to increase in strength. Of course, you may think, well, but if the strength of cis-regulator increases and increases, perhaps it pushes expression levels up, and perhaps that's not such a good thing. If you got a stronger cis-regulator, perhaps you produce too many proteins, and perhaps that's not a good thing. And in fact, when you model this, so you introduce the idea that if you depart from an optimal quantity of protein produced, you got a fitness penalty. So that's the optimal production of a protein, and that's the expression you achieve. If it's too large, then it's not good. If it's too low, it's not good either. And you can introduce other loci that contribute to total expression. So I will just give you a very brief example where the second locus is another cis-regulatory sequence. In that case, I'll just jump here. So you introduce the first cis-regulator at that point here. So it reaches some value, and it stops because expression level increased too much and is deleterious. But at that point, you introduce mutation on the second locus, and what's going on is that they each evolve in combination to compensate each other. Okay? So you got coevolution of all the regulators together, such that the expression level stays the same, yet you've got permanent and very quick evolution of all those sequences. So that's a summary of part 2. If there is diploidy, if deleterious mutations are partially recessive, then cis-regulator evolve in a runaway fashion. That triggers coevolution of all the regulators. And so regulatory complexity emerges selectively and not because of individual advantage here. So we are done with that slide. So I jump to my conclusions. First, this theory, in fact, I didn't have time to explain all the other theories which are around about the evolution of evolutionary complexity, most of which can be summarized by saying that complexity is more or less maladaptive. It's just arising in small population because of the fixation of deleterious mutations. So this theory here is not at all about this fixation, this maladaptive complexity. It's not really an adaptive in the Darwinian sense that you've got an optimization of some individual level properties. But it's Darwinian, it's positive, you've got selection here just because you've got genes that are favored and that spread because of natural selection. So it associates diploidy and regulatory complexity explicitly, explaining perhaps better why you find this regulatory complexity in eukaryotes and not in prokaryotes, which are haploid. And it emphasizes genetic conflicts as a major engine, of evolution, rather than just chance, as you could imagine. So, like in evolutionary biology, we often use a metaphor of the Red Queen, taken from Lewis Carroll's book, where now here you see it takes all the running you can do to keep in the same place. Says this Red Queen to Alice, saying that you've got to run faster, just stay in the same place. Or if you prefer, in movies, everything needs to change so everything can stay the same idea, that you've got here a lot of evolution which is going on, but on the surface, at the individual level, you barely notice. So if I can sketch a very quick history of early eukaryotic evolution. So you first had this first gene controlled by a cis-regulatory region. Because of the process I was explaining, it increases in strength, it multiplies. Got a lot of complexity in the cis-regulatory region emerging. At some point, you lack some transcription factors. There are not enough of them to go further. Then perhaps you mutate here to get some new, better strengths of the cis-regulatory region. And that's the point where you got the evolution of this overwired regulation in eukaryotes. So we are, like 1 billion years ago. Of course, then you can have all sorts of consequences of those processes. And for instance, you can imagine that the whole region here is duplicated. And now you got the two versions of that gene. But because you got such a complexity in the regulatory region, it's fairly easy to evolve, for instance, tissue-specific expression, where you just switch off different cis-regulator in different tissue, so that you got in multicellular organisms you can evolve different tissue and what is called development. All right, so that's what I wanted to say. I've been explaining those different arrows. I'm working on some more integrated project here at Radcliffe. Didn't have time to talk about everything. And I finished by quoting about, of course, Wittgenstein. I think that you can see that genetic conflicts can generate a lot of complexity and a lot of multiplicity that you would not expect if you just think in terms of a kind of naive Darwinian view of evolution. Of course, it's more difficult to disagree with Shakespeare. And I've just put in green the parts where I think you can tweak a little what he said. Thank you.